Welcome once again to Population Genetics and Evolution, Unit 1, Session 5, the Hardy-Weinberg Equilibrium. And by the end of this session, you should be able to explain the Hardy-Weinberg principle and understand and apply the Hardy-Weinberg principle in population genetics. One of the more difficult topics for introductory biology students to understand and for teachers to teach is the Hardy-Weinberg Equilibrium principle. One reason for this difficulty is the student's mathematical background. More problematic than lack of manipulative skill is the difficulty of understanding why the principle is true and understanding why the principle applies to specific populations, or more importantly, the value of its application. The Hardy-Weinberg principle or equilibrium is, of course, the cornerstone of introductory population genetics and is therefore an important part of understanding evolution as recognized in most science standards documents. For example, adaptation by natural selection is one of the core ideas in the next generation science standards, focusing on how the distribution of traits in a population changes. Likewise, the next generation science standards targets the ability to use mathematical representations to support scientific conclusions and design solutions. Students who demonstrate understanding can use mathematical representations to support explanations of how natural selection may lead to increases and decreases of specific traits in populations over time. Given its importance and the well-recognized teaching and learning difficulty in accord with these standards, in this book, we explain basic concepts of the Hardy-Weinberg equilibrium, emphasizing distinctions that are sometimes ignored at the cost of coherent understanding. In general, a system is said to be in equilibrium if all competing influences are balanced. In the body, for example, we speak of homeostasis as the ability to maintain the internal equilibrium regardless of changes in the environment, example, temperature. A basic precept of evolution is that under certain conditions, the frequencies of genotypes and therefore of alleles do not change. They remain at equilibrium, but in the absence of these conditions, the frequencies do change. Definition: a population is in Hardy-Weinberg equilibrium is if the genotype frequencies are the same in each generation. Consider the simplest situation of a monogenic Mendelian trait, a pair of alleles, one dominant A and the other recessive A, within a population of n individuals. The frequency of the dominant allele is the number of alleles divided by the total number of alleles at this locus within the population, 2 times the number of individuals. For example, if n is 4,000 and 2,000 of the alleles of this locus in a population are recessive A, the frequency of the dominant A allele is 3/4, and that one of the recessive allele, that is remaining non-allele A, is therefore 1/4. These therefore would be the allele frequencies if there are 1,000 recessive AAs and 3,000 dominant AA individuals, or 500 recessive individuals and 1,000 co-dominants and 2,500 dominant. But if these individuals randomly mate, the next generation has the same allele frequency but a genotype frequency of 1/16 AA, 3/8 AA, and 9/16 AA. If a final population is at H minus Q equilibrium, however, both the genotype and the allele frequencies will be essentially the same in subsequent generations. We will demonstrate that a population is in Hardy-Weinberg equilibrium if the following conditions hold with respect to a particular gene. There is no migration in or out of the population. Natural selection is not occurring. Mutation is not occurring. Each member of the population is equally likely to breed. The population is infinitely large. Full random mating, thus each pair from the population is equally likely to breed. Females of a species often prefer males with certain traits. Examples originally identified by Darwin include peacock feather displays, antlers in deers, and the manes of lions. Observation 1: as long as a population satisfies biological conditions 1 to 6, the allele frequencies P and Q are are the same in each generation. So why is this so? Conditions 1 and 2 guarantee that there is no change in the allele frequencies between the birth and maturity of the next generation. There are no unaccounted forces that would change the allele frequencies. That is, one phenotype is not more fit than the other. Conditions 3 and 4 guarantee that at birth, the pool of alleles in the next generation is the same as in the current generation. Mating just reshuffles the alleles. The allele frequencies remain the same. The population needs to be finite to guarantee that the frequencies remain exactly P and Q. The probabilities P and Q represent the averages over many trials, and so it will only be approximate in a particular trial on a finite population. A change in allele frequencies can be caused by genetic drift or a bottleneck. Of course, no population is truly infinite, therefore condition 5 can never be strictly met if a population is large enough. However, it is considered to be effectively infinite. Likewise, the other assumptions are rarely, if ever, true of a given population. Example, the mutation rate is rarely zero. Heidelberg equilibrium is largely a theoretical state like a frictionless plane, an absolute vacuum, or travel at the speed of light. As with those concepts in physics, it nonetheless plays a fundamental conceptual role in biology and is a valuable tool for understanding evolution. The HW principle, Hardy-Weinberg principle, has many applications in the modern practice of evolutionary biology, where its value often lies in identifying when HWQ does not exist and when determining which factor or recombination of factors most likely explains the observed change in allele or phenotype frequencies over time. That is, what are the drivers of evolution in this population? Also, the evolution neutral mutations. The population is often close enough to equilibrium to provide a tool for comparing their frequencies against the frequencies of linked genes of interest to understand how close the matter are to the Hardy-Weinberg equilibrium. The following textbook problems are built on the assumption that if a population is in Hardy-Weinberg equilibrium, which is often a dubious assumption, then it is possible to calculate the allele frequencies from the frequency of the homozygous recessive, which can be found by observation. That is, under the assumption of HQ equilibrium, if B% of the population is homozygous, then Q equals to B, so Q equals the square root of B. And because P equals 1 minus Q, then P equals 1 minus square root of B. Problem 1. The data below demonstrates the frequency of T-test and non-T-test in an isolated population at Hardy-Weinberg equilibrium. The allele for non-T-test is recessive. How many of the T-tests in the population are heterozygous for testing? T-tests are 8,235. Non-T-tests are 4,325. Solution provided. An acceptable answer would be any number in the range of 6,030 to 6,156, depending on how the students rounded the variables in the Hardy-Weinberg equation. The comment on problem 1. This is a standard HQ equilibrium problem. The frequency of non-T-tests into bracket homozygous recessive individuals Q2 is 4,325 over 1,200 over 12,560. Assuming that the population is in Hardy-Weinberg equilibrium, the frequency of the homozygous recessive allele is computed as the square root of the frequency of homozygous individuals Q2, giving us Q equals the square root 4 into bracket 4,325 over 12,560 bracket close. Therefore, Q equals 0.58 and P equals 1 minus Q equals 0.42. Then the frequency of the heterozygote 2 PQ equals 2 into bracket 0.58 and then bracket 0.42, giving us 0.49. This yields the number of heterozygotes as 49 by 12,560, giving us 6,119. Problem 2, the ability to taste phenylthiocarbamide (PTC) is due to a single dominant allele T. You sampled 250 individuals and determined that 150 could detect the bitter taste of PTC and 65 could not. Calculate the following frequencies: the frequency of the recessive allele, the frequency of the dominant allele, the frequency of the heterozygous individuals. The frequency of the recessive allele, Q equals 65 over 2112, Q2 equals 0.35, then Q equals the square root of 0.30, Q equals 0.55. Question 2, the frequency of the dominant allele, P equals 1 minus Q, giving us 1 minus 0.55, P equals 0.45. Then 3, the frequency of the heterozygous individuals. Let X equals frequency of the heterozygous individual equals 2PQ, X equals 2 into bracket 0.45 by 0.55, bracket close equals 0.495. Comments on problems 1 and 2. Both problems focus on making calculations that students can do without understanding what H and Q equilibrium is. In problem 1, H-Q equilibrium is explicitly assumed so that the problem is technically correct, but it doesn't say what chemical was being tested, presumably PTC, so it doesn't ask students whether HQ conditions could be met for this trait, and students could only solve problem 2 by assuming Hardy-Weinberg equilibrium, which is not justified. The ability to taste PTC, a beta substance that cannot be tasted by some individuals, is frequently used in HQ equilibrium problems, likely because it is assumed to be selected neither for nor against, because PTC does not occur in nature. Thus, the student is expected to deduce or more likely assume that the HW conditions apply. Teachers and textbooks, however, rarely make this reasoning explicit, leaving students with a misperception that understanding PTC testing is just a game or a puzzle, which likely seems unimportant to them because it doesn't relate to their daily lives. In fact, recent research has shown that the ability to taste PTC is strongly correlated with the ability to taste other beta substances that do occur naturally, many of which are toxins. In fact, humans have about 30 genes that code for beta taste receptors, allowing people to taste a wide variety of beta substances. Genetic Science Learning Center 2014 made this research. Thus, it seems likely that the ability to taste beta substances such as PTC is positively selected for. A second issue that arises in problems about sickle cell anemia is that two opposing selective pressures are at work: a positive selection for heterozygosity and a negative selection against affected homozygotes. Such a situation can produce balanced polymorphism equilibrium of alleles, but not HW equilibrium, because the genotype frequency will change. This makes sickle cell anemia a poor choice for the context of most introductory level HW problems. A third objection to this entire analysis of sickle cell anemia is that Hardy-Weinberg analysis requires addressing the idea of a single generation. This is a very difficult concept to apply to human populations without careful data collection, because people living at any one time can represent 3 or 4 generations. Problem 3, people who are homozygous for having 2 copies of a certain 32 BP deletion mutation in a gene known as CCR5 are known to be largely resistant to HIV infection. CCR5 is the main co-acceptor molecule that allows the virus to attach to certain white blood cells and enter them, thereby establishing an infection. In a study of 1,380 random Caucasians of childbearing age in the United States, 1,102 individuals were found to be homozygotes free of this deletion, assumed that the US Caucasian population is at Hardy-Weinberg equilibrium at this locus. Susan, a Caucasian American woman, at increased risk of HIV infection because she has multiple sex partners. What is the probability that Susan has little reason to worry about HIV infection? That is, she is homozygous for the deletion. What is the predicted frequency of US Caucasians of this age who are carriers of the prospective deletion? How many heterozygotes will be expected in this sample? Before HIV appeared, would you have expected the population to have been at HW equilibrium at this locus? Why or why not? State your assumptions. In the absence of effective HIV treatment, what would you expect to happen to the allele frequencies over time? How would you expect the allele frequencies to change over time once effective HIV treatment was in use? Why? How would your answer change if the HIV treatment was only effective for people past childbearing age? Answers to questions 1 and 3. Let P be the frequency of the allele without the deletion. P2 equals 1102 over 1318 equals 836. Since we assume HW equilibrium, P equals 0.836, the square root of 0.836 given as 0.914. Since there are only two alleles, Q, the frequency of the allele with the deletion satisfied Q equals 1 minus P equals 1 minus 0.914 equals 0.086. Q2 equals 2 equals 0.007. So Susan has 0.07% chance of being at lower risk. Again, assuming the HW equilibrium, 2pq equals 0.914 bracket close bracket open 0.086 giving us 0.157 times 1318 giving us 207. Before HIV appeared, the population was likely at HW equilibrium at this locus. This assumes that the deletion was selection neutral. With the appearance of HIV and in the absence of effective treatment, we would expect that the frequency of people without the homozygote deletion P equals 2 and 2 PQ should decrease, shifting the population out of Hardy-Weinberg equilibrium. The advent of an effective treatment should move the allele frequency distribution back toward equilibrium. A treatment that only has an effect after childbearing age would have effects on the allele distribution similar to no treatment, because for a mutation to have a natural selection effect, it must affect reproductive success. In review, the Hardy-Weinberg equilibrium is of the cornerstone of introductory population genetics and is therefore an important part of understanding evolution as recognized in most science standard documents. A system is said to be in equilibrium if all competing influences are balanced. In the body, for example, we speak of homeostasis, as mentioned earlier, as the ability to maintain the internal equilibrium regardless of changes in the environment, example, temperature. The basic precept of evolution is that under certain conditions, the frequencies of genotypes and therefore of alleles do not change. They remain at equilibrium. But in the absence of these conditions, the frequencies do change. A population is in Hardy-Weinberg equilibrium if the genotype frequencies are the same in each generation. A population is in Hardy-Weinberg equilibrium if the following conditions hold with respect to a particular gene. There's no migration, gene flow, or in out of the population. Natural selection is not occurring. Mutation is not occurring. Each member of the population is equally likely to breed. The population is infinitely large. For random mating, that each pair from the population is likely to breed. Females of a species often prefer males with certain traits. Example, originally identified by Darwin include peacock feather displays, antlers in deer, and the males of lions.